Hello everyone, welcome to KR Microbiology classes and this is K.Rajani DL in Microbiology, Telangana Tribal Welfare Residential Degree College Khammam. Today I am going to explain about IIT JAM 2025 question paper, especially focusing on the DNA related topics. Okay, so let's move to the topics. So first question, after how many cycles of polymerase chain reaction, the amplified product of required length is generated for the first time from a chromosomal DNA template? Here, the question is about the first appearance of the required length DNA. So, for this, you have to remember that in the first cycle, we will get the DNA length, we will get the DNA which has the more length than the required DNA. And the second cycle, whatever the unnecessary regions they cut down, and we will get the uh, intermediate length DNA, and in the third cycle we will get the first time exact length DNA in that PCR. So answer is the third cycle. Next question, which of the following activity is not possessed by the human immunodeficiency virus 1 reverse transcriptase? So here the question is, what are the functions of the reverse transcriptase? In this case you have to understood that synthesis of DNA from RNA, obviously, it will happen in the HIV virus, and synthesis of DNA from DNA. So, whenever the RNA produces one strand of DNA, that DNA will produce another strand. So, synthesis of DNA from DNA will also happen. And after preparation of the one DNA strand from the RNA, that RNA will be degraded. That RNA will be degraded, and this single DNA will forms the another strand of the DNA, right? So in that purpose, as mentioned in the C point, degradation of RNA strand of RNA is also happened, right? So what is the third one? Synthesis of mRNA from DNA. This was not carried by the reverse transcriptase. So the wrong answer is the wrong work by the reverse transcriptase, but it is only the right answer, right? So synthesis of mRNA from DNA is the not work of the reverse transcriptase. So whenever you are attempting a question, please read the question mindfully, because here they are asking which is not possessed by the particular enzyme, right? So this is the answer D. Next question. Mechanism of antibacterial action of polymyxins relies on. So polymyxins acts on which part of the bacteria they are asking, right? What is the mechanism of the antibacterial action? Here, see the four options: inhibition of 30S ribosomal subunits, disruption of peptidoglycan synthesis, inhibition of DNA replication of bacteria, and disruption of the membrane architecture of the bacteria. Here, you have to read, like whenever you are preparing for the exams, you must have to understand different kinds of antibacterial materials or toxins or antibiotics, and on which part they will specially act. Okay, so in this case, polymyxins acts on the lipopolysaccharides, especially lipid A. It acts on the lipopolysaccharides, and it increases the permeability of the cell wall of the gram negative bacteria. So here the correct answer is the disruption of the membrane architecture of the bacteria. In the next question, if you see here, which one of the following statement is correct about the agrobacterium tumefaciens mediated plant transformation? Sorry if I am pronouncing something differently, that's okay. You can understand, I hope. Which one of the following statement is correct about agrobacterium tumefaciens mediated plant transformation? So you all know very well that agrobacterium tumefaciens is a very good plant transforming bacteria, because it contains plasmid, right? It contains a ti plasmid which is capable of transforming the required genes required genes to the plant cells in a very standard manner. So it can include that bacterial DNA, I mean that plasmid DNA, into the plant cell DNA. That's why scientists uses the agrobacterium tumefaciens as a very good tool to transform any required genes to the plant cells. So here see that first of all, before to the selection of the correct answers, you just remind that this TA plasmid contains what they will contains, right? So transfer DNA and virulence genes. Transfer DNA contains it contains a right end and left end, and it also contains the virulence genes, which in which place we will include the required selected genes that has to be placed in the plant cells, right? So now see the options. Option A, the marker gene for the selection of plant transformant is located outside the tDNA segment of the transformation plasmid. The marker gene of the selection of bacterial transformant is located within the tDNA segment of the transformation plasmid. Two DNA border sequences left and right are essential to design a binary vector system. In a binary vector system, the virgins and two DNA border sequences left and right are present in the same transformation plasmid. So these are the four options, right? So this question is multiple answer question. This is because you can select more than one answer. So see here, the marker gene for the selection of bacterial transformant is located within the tDNA segment of the transformation plasmid. Option B is correct and option C is correct, because the marker genes for the selection of bacterial transformant is located within the tDNA only. Then only it can be transformated, right? And the second option, if you see that I mean C, option C, two DNA border sequences are essential to design a binary vector system. Yes, obviously, tDNA, which contains the left and right border sequences, is very important to transfer any DNA. So here, option B and option C are the correct answers. If you see the next question, this method is used to detect. If you see this question, this method is used to detect a DNA fragment of 150 base pairs are. So which method is used to detect the DNA fragment of 150 base pairs? Here you have to remember always one thing. 150 base pairs means it is a very small DNA fragment. Okay, 100 to 500 up to 500 base pairs means it is a very small DNA fragment. Above 500 means it is a moderate size of the DNA fragment. So in the case of a small DNA fragment means very small number of base pairs also, there are some particular processes are there. So here they gave four options, right? Option A, agarose gel electrophoresis. Option B, northern blotting. Option C, polyacrylate gel electrophoresis. And option D, western blotting. In this case, agarose gel electrophoresis and polyacrylate gel electrophoresis. Usually we will call it as PAGE, right? Polyacrylamide gel electrophoresis. These two are the right options. And then what about the two wrong answers? So northern blotting technique is used for the detecting of the RNA, and this western blotting is used for the detecting of the proteins. So correct answer is option A and option C. Okay, so let's move to the next question. So this is a somewhat very big question. It will take some time to just understand the question only. So read it carefully. A cDNA was synthesized from the mRNA of a eukaryotic gene after cloning and sequence analysis. The double-stranded cDNA of 614 base pairs revealed 125 base pairs 5' UTR and 120 base pairs 3' UTR. The number of amino acids present in the polypeptide encoded by this gene is. Here the question is, please carefully read it again. Take some time, pause the video and just read the question again and again. Okay. So here what he is telling, there is a double stranded DNA. It has 614 base pairs. Okay. And in that 614 base pairs, 125 5' base pairs and 121 123 base pairs, they are untranslated. Okay, so they are untranslated or untranscripted regions. They are untranscripted regions, so they don't involve in the protein synthesis, right? I mean, they don't code any amino acids. That's why they are called as the UTR, untranscripted regions. So now what they are asking, the number of amino acids present in that polypeptide which was encoded by this gene means total number of genes are less than the 614, because in that 614, 125 5 prime and 123 prime, they are not participating in the coding, right? So here they are not asking how many base pairs. Carefully observe the question. They are asking how many amino acids will be present. So this is little bit tricky, just only little bit tricky. This is very little bit tricky. See here, given data, total double stranded cDNA length is 614 base pairs, 5 prime UTR 125 base pairs, 3 prime UTR 120 base pairs, right? So now see here, the coding sequence length, how much it is. So we have to deduct the UTR sequence base pairs, right? So 614 minus 245, that is the 369 base pairs which are participating in the amino acids coding. Out of 369, you have to remember that there are there is one stop codon will be there, right? So there is a sorry. Now I have to tell you here 369 base pairs are there, right? So they are asked how many amino acids are participating, right? How many amino acids? Here, one codon, one codon, it one codon gives, I mean, it gives the one amino acid, right? One codon gives the one amino acid. It encodes the one amino acid. So see here, one codon means three base pairs. So 369 by 3 means 123 codons. Total 123 codons are there in these 369 base pairs. Out of 123 codons, there is one stop codon will be there. It don't encodes any amino acid, right? So total how many finally? 123 minus 1 stop codon. Total 122 codons means 122 amino acids are participated in this polypeptide chain. So little bit tricky. So once you observe and once you understand what is the core concept of that particular question, you can answer very easily. Thanks for watching, and if you have any doubts in any topics, please mention in the comment section. Thank you. Thank you very much.